pleasure to have Fedor Jelezko, Professor Jelezko, today here with us, right? So Fedor, well, he has a very long, outstanding career in quantum technologies, right? So he's currently director of the Institute of Quantum Optics in Ulm, right, at Ulm University, as well as the director of the Center for Integrated Quantum Science and Technology, a member of Heidelberg Academy of Sciences, right? So he did the PhD in Belarus, right? And then moved to Stuttgart with Professor Brachtrup, right? Yeah. And well, he has many results actually in this field, but not only in academia, right? Because Fedor, well, he's also involved in the development of quantum technologies in different companies, in particular Envision Imaging and Diatope, both in Ulm, right? So thanks a lot for coming, Fedor. It's really a pleasure, and the stage is yours.
Yeah, thank you very much, George. And I would like to thank Quantum Center for invitation. It's a pleasure to be here, to see the development here in Basque Country, especially in Bilbao, and rapidly growing research related to quantum technologies. And I would like to share today our work on diamond spin qubits. As was mentioned by George, so we are at the center of integrated quantum science and technology. It's our Southwest Germany competence center in quantum science, and I think there are many opportunities also to link our research and to have this very strong pillar here in Basque Country. It's very exciting, and I'm sure that there will be connections not via spin qubits in diamond only, but also in wider range of research between our quantum centers. But today we'll talk mostly about this particular type of qubits, dopants in diamonds. They are a bit special in terms of combination of pulses coming from being a solid state system, but also having very long coherence time, similar to coherence time of ions in traps or neutral atoms. Here we have a solid state atom trapped in a diamond lattice, and the diamond lattice is placed in kind of the very best vacuum chamber, so the dopant is trapped very well. At the same time, you can bring it to region of interest if you do sensing, or you can place many of these color centers together in a very short distance if you like to do conditional dynamics. And there is an interest now in a community for all these applications. Actually, there are even companies trying to build NV center in diamond, color center in diamond-based quantum computers and simulators. And there are applications in sensing, especially for nuclear magnetic resonance signal with very high spatial resolution. There are even some applications related to medical imaging. And so all this comes from a fundamental quantum optics experiment, but now there's a variety of applications that are under exploration. So today, owing to limited time, I will only touch this sensing part. And so saying about sensing maybe is one of the most promising avenues of application in quantum technology, often a little bit overlooked. We all know that quantum computers hold a very big promise to, make computations exponentially faster compared to classical computers, but it's still a field which needs to be developed over the next decades. You need very large number of qubits to explore quantum coherence to reach the regime of quantum advantage. But in metrology and sensing, we already now have a number of qubits that reaches regime where you can do better than with classical devices. That holds for clocks. The best clocks are using long coherence time of atomic transitions, and that holds for sensors too. And so today I would like to share with you how you can use spin qubits in diamond, long coherence time, and in some cases also more than one qubit dynamics in order to realize quantum magnetometers. the system, the qubits that we are using is nitrogen vacancy color center in diamond. Actually, there are hundreds of known dopants in diamond which are optically active, but this nitrogen vacancy color center attracted attention because it combines several very interesting, very promising spin and optical properties. From the structure, it consists of nitrogen sitting at the lattice position plus vacancy nearby. And this vacancy in diamond is not really a vacuum. It means that there are some dangling bonds which are free. In total, this system has 4 dangling bonds electron, 1 additional electron from nitrogen, and 1 coming from external donor to form a 6 electron system, like artificial molecule in diamond, having strong optical transition. When present in a large concentration, this color center give a deep pink color to diamond. And this also tells you that you can excite this color center with laser light in a visible range. That's why you have a color, and it also can emit photons. Now the next point comes, so this optical excitation and emission leads to polarization of the spin state of this color center. Here I show you a little bit more details about structure, energy level structure of the color center. It consists of ground state, electron spin triplet, and excited state triplet. And when you excite this color center owing to passage through a singlet state, which is spin selective, the system falls down with very high probability in one particular sublevel of the triplet state spin manifold in so-called magnetic spin quantum number 0. This magnetic quantum number 0 substate fluoresces stronger if you excite it with laser compared to 2 other sublevels. And by measuring fluorescence intensity, you can distinguish between 2 spin states, or one of spin state, ms equals 0, and 2 others. These plus and minus 1 spin states have a lower fluorescence intensity compared to ms equal 0 spin state. So if you do experiments when you shine a laser on your system, here you switch the laser on and you measure fluorescence intensity, the response of NV center in terms of number of emitted photon will be different for ms equals 0 spin state, which is darker here in this line, compared to ms equals 0. So ms equal plus minus 1 are darker compared to ms equal 0. So this provides you a way to measure the spin state with very, very high efficiency, because optical technique of measurement can be very effective. So you have very good detectors typically, and so measuring spins using emitted photons is very efficient compared to measuring spins using conventional nuclear magnetic resonance and electron paramagnetic resonance coils. So still, usually we don't collect all the photons, and experiments in order to get information about single spin need to be repeated a few 100 times. The NV center is also more than one qubit in itself. There is also a nitrogen atom involved in this 6 electron system, and nitrogen do carry nuclear spin. That's why if you look carefully on the transition of an electron spin of NV center, you do see 3 sublevels here present, and these 3 sublevels correspond to different eigenstate of nitrogen nuclear spins. So these nuclear spins can be used as a resource. It has a long leaving spin state, long compared to electron spins. And in principle, you can also map the state of nuclear spin to the state of electron spin and then measure it optically by using kind of quantum gate type operations. And if you do this, you can also get access to nuclear spin state of color center. So basically making a swap operation between electron spin and nuclear spin, and then measuring electron spin, then you can repeat it many, many times. And in this case, you can observe dynamics of a single nuclear spins. And because of longer coherence time and longer relaxation time, that's allow you to observe quantum jumps. What you see here is the fluorescence intensity versus time, measured for a single in V center. And what you see, there are jumps between bright and dark states. And these jumps now correspond to jumps of a single nuclear spin between spin up and spin down. And the time scale here is about 1 second for the whole acquisition time. So that's very slow dynamics, and it's allow you quantum non-demolition measurements of a single nuclear spin by measuring now this intensity of fluorescence. So every time you measure bright state here, it's a nuclear spin up. If you have your NV center in a dark state or darker state, then it's nuclear spin down. So it's very interesting because it can be now done in ambient conditions. So there is no vacuum here, and NV centers can be also placed very close to the object of interest. But this readout is a very important element of all the other experiments that we are going to discuss. Now, from technological perspective, it's also very important to be able to create these color centers at well-defined locations. And so here, effort of material science enabling generation of color centers with high accuracy was very crucial. And so the success of the field is based on a shoulder of material science success, which enabled all the other experiments. It was shown that, first of all, you can grow diamond of very high purity where there is no background color centers. So that's material having no parasitic qubits is very essential starting step. And starting from this material, one can create color center by implantation of single nitrogen ions using focused ion beams, or if you want to improve the spatial resolution better than focus of the beam, you can use nano apertures like holes in the IFM tip, or nanostructures created by electron beam lithography. In all these cases, position accuracy for these arrays or position of single NV centers is on the order of 10 nanometers. So you can place these qubits with a precision which is of molecular scale. And so this is now interesting in terms of sensing, if you want to sense single molecules, or it can be interesting in terms of creating quantum computers, because at 10 nanometer distance, the interaction between two single electron spin is not negligible anymore. It can be stronger than any environmental induced decoherence here in the system. I'll give you a few insight about experiment. It's a very simple compared to many other platform setup. Detection itself is based on a confocal microscope where we focus light tightly to diffraction limit, and we scan the excitation spot through the sample. Every time we hit NV center with laser light, we get fluorescence. We detect with sensitive avalanche photodiode. We can also check whether these bright spots are really single color centers by measuring G2 function of emitted light and photon anti-bunching present here at zero delay time of G2 function is a sign that emitted photons are really coming from a single quantum system. They're emitting photon one by one, and that gives you then guarantees that you work really with a single qubit. In addition to optical setup, which is very conventional confocal microscope, we also apply microwave to control the spin state using microwave antenna. And now by measuring brightness of a single color center and sweeping the microwave through the resonance, we can observe spin flips if we hit the right transitions, and that lead to redistribution of population between different spin sublevels. Remember, they have different brightness, so you get a drop in fluorescence by 30% if you hit the the spin transition. And these experiments can be now done because it's optically detected on a single spin level. So you see that it's a single atom by measuring G2 function, but you have quite a significant signal-to-noise ratio already after a few tens of second acquisition time on detecting the spin state of these color centers. This is a scheme of experiment. I show you also heart of this experiment in photo here on the left. It's a piece of diamond where we deposit microwave antenna. It's not a resonant antenna. It's not like emitting also microwave. It's actually near field effect. It's, we are passing microwave current here through. The distance here between this narrow part of waveguide is about 10 to 50 micrometers, and EHU centers that are used in these experiments are implanted here in this region. So the microscope lens is not shown here. It's coming here from the top, and the photons that are collected now are plotted versus, for example, application of microwave pulse lens. And what you see here, example of Rabi oscillations, again measured optically. We measure the population of bright state of EHU center, and you see that this population oscillates. That's give you like a very basic coherent control experiments driving a single spin using microwave, and it's also give you now the time scale of this drive. So it can be done on with a Rabi frequencies which are reaching a few tens of megahertz. So you need just a few nanoseconds to flip the single spin. All right, so now after this introduction, I would like to highlight a few experiments to you. And today I will talk about quantum sensing from perspective of limits of its sensitivity. There are some fundamental limits related to sensitivity and also spectral resolution. Then I will show you a few examples of new developments in nanoscale nuclear magnetic resonance, using nanodiamonds for sensing of signal inside of living cells, and also will show you how you can use spins to measure temperature with nanoscale spatial resolution. And if I will have time, also will talk a little bit about magnetic resonance imaging, medical imaging applications of these qubits. And I will start with now converting qubit to a magnetometer. So for qubit, usually you like to have a very long coherence time, but also not to be very sensitive to external perturbation. But spins are intrinsically coupled to a magnetic field. For a spin half system, you have Zeeman splitting. The same true for spin 1 system, which is in V center. So if you apply a magnetic field, one of the sublevels is not reacting on the field because it's magnetic quantum number 0, but two others are getting splitted. And if you measure the splitting, it's related to magnetic moment of a single electron, which is very well defined. So by measuring the frequency splitting, you can measure magnetic field. And so this is Zeeman effect-based magnetometer. This is experimental data here. You see two transitions associated to 0 to +1 and 0 to -1 state. You measure the frequency and then you calculate the magnetic field. So what about the sensitivity of this approach? Well, sensitivity of tracking the frequencies is related to noise present in the system. Here it's a photon number, it's always noisy, but also to width of the transitions. The sharper is the transition, the better you can track a frequency. It's very intuitive. You can also put it in more precise way of definition of limits, and if you do analysis, so you can get so-called standard quantum limit or spin short noise limit in this magnetometry. So the experiment we do here for magnetometry as a model is exciting coherent state of a spin, a superposition of spin up and spin down. This state precesses in external magnetic field, and after some precession time, you measure the phase. The phase is proportional to the magnetic field. So if you know the time, you can measure the magnetic field. But there is always intrinsic noise, and there is also limited time you can acquire this phase. And the smallest possible magnetic field you can measure. This is sensitivity per definition. Now it depends on the parameters of the atom, the magnetic moment of the electron, but also the number of atoms, total measurement time, and coherence time t. Those are parameters that define the sensitivity, provided that you don't have any other technical noise sources. Now, if you want to improve the sensitivity of this magnetometer, there are several ways, but not so many ways. So you don't change so much parameter of the atoms, so you need to play with this term. So you can increase the number of atoms, having more NV centers contributing to the signal. That reduces the noise, having more copies of qubits which give you the signal. But that creates a larger volume of the sensor, and so there is an interesting avenue here which I'm not going to discuss today. So I will fix this n to be one single atom as a sensor, and then the rest is just measurement time and coherence time. So the only parameter that helps you in increasing the sensitivity for a given measurement time is the coherence time. You need to acquire the phase for longer to reduce the noise. That's true for actually any quantum sensor in case if it's limited by the projection noise. And for NV center in diamond, so this T2 time is quite long. Actually, here is an example of this experiment. I excite coherent state of NV center, superposition 0 and 1, and leave it to precess under ambient condition. And I measure now this precession by interrupting this precession and measuring the state again at the end. And you see that this precession leaves here for a few hundreds of microseconds. So a few hundreds of microseconds of T2 time, it's an important resource, especially if you think about ambient condition applications. Not many qubits, and I would say no other qubit, can have this long coherence time if you're talking about electron spins. Nuclear spins can have longer coherence time under ambient conditions, but for electron spin qubits, this is the record value. Now, you can also do more with these magnetometers. You can also make a step from a DC magnetometer to a spectrum analyzer. And this is typically done by applying many microwave pulses, kicking your electron spin forth and back between eigenstate. This train of microwave pulses lead to refocusing of static noise and keeping your qubit to be sensitive to particular frequency. So when you now measure the sensitivity function, or you plot the sensitivity function of qubit, it will have sharp peaks in frequency domain, and this type of application of train of microwave pulses allow you to tune your magnetometer to be sensitive only to one particular frequency of AC fields. It's a bit similar to optical spectrometer, where you have a grating and you get a positive interference of light of particular frequency in one direction, so phase matching. So the same is happening here. There's a time domain, and there is only one frequency which is kind of allow phase matching and accumulation of phase in a constructive way, and all other contributions, all other frequencies are getting refocused. And so the analogy with the optical spectrometer is not very bad. Actually, the more pulses you apply here, the narrower get your frequency response, so similar to number of traces in optical spectrometer. So this is an important instrument for spectroscopy using single qubits, because you now can also tune your spectral response. An example of applications, there are multiples, but I think one of the most promising one is to detect magnetic resonance signals coming from spins outside of diamond. I give you one example here. It's in V center, which is implanted below the surface with this precision of a few nanometers I mentioned before, and the biomolecules was attached to the surface of diamond just above. That was ubiquitin, a stable protein, and this ubiquitin structure is in principle known, but we just wanted to detect the presence of the molecule via dipole-dipole coupling with NV electron spin, or if you want, by hearing the Lama precession frequency of nuclear spin present in a protein and using NV center spectrum analyzer to detect the magnetic field of this nuclear spins outside. And this is a collaboration with Michael Lukin, Hong Kong Park, and Ron Walsworth in Harvard. I give you a little bit of examples of how you really can interface now qubit with biological molecules. It's maybe not trivial, so you have to now bring these molecules at the very top of NV center, and this is done by modifying the surface of diamond. Diamond is very inert, but you can start with a carboxy group and via amino group make a specific chemical reaction with a protein. The protein binds now covalently to the surface of diamond, sits there stably, not moving so much. And so here on the right side you see IFM pictures of diamond showing very flat topography before attachment of molecules, and a rough surface with a nanoscale kind of roughness, typically corresponding to size of this protein after chemical attachment of the protein. So you can create close to a monolayer of this molecule, and you can now think, well, maybe it's not single molecule detection, but remember that NV center sits so close to the surface that it sees only one of these molecules, only one of these molecules, even less than a whole molecule. Only a fragment of the molecule will be seen by a single NV center. And here, an example of spectral lines. This is now a noise, magnetic noise that NV center feels, that it detects, and we can scan the frequency of sensitivity by tuning this train of pulses and make a kind of a spectral scan, and you see that this environment of NV center do have peaks, spectral lines associated with Lama precession frequency of hydrogen present in a protein and carbon present in a protein. It's a signal coming from a single biomolecule. Just to give you a full what is improvement compared to state of the art. A typical nuclear magnetic resonance experiment is done on billions of molecules. So here, by approaching a sensor to a single molecule, you can be in orders of magnitude in the sensitivity. However, you see that spectra are noisy, although they've been acquired for tens of minutes. And line width, which is related to this time interval between pulses, is not very sharp. It is not yet good enough, this experiment, to answer the question about the structure and dynamic of the molecule. And so the big effort now here is to come from the detection to understanding the chemical structure of molecule or its dynamics. There are some interesting developments here. There's a demonstration of detection of a chemical shift inside of a liquid sample by a Krachtrup group some time ago. You see the peaks associated with fluor sitting on the same molecule but having a different chemical environment. Again, spectra are noisy and not super well resolved. And so the big picture goal here in this field is how to improve it and make spectra less noisy, reach resolution which is better than Hertz to understand kind of capture dynamics of big molecules. Usually it happens in the millisecond time scale or hundreds of milliseconds. And also reach spectral resolution which allow to understand interactions between nuclear spins inside of molecules. For conventional NMR, this high-resolution spectroscopy was a success story, allowing to unravel dynamics of proteins and unravel structure of proteins. Here is an example of myoglobin protein spectrum obtained by conventional NMR. You see very large number of sharp spectral lines, and so understanding the splitting between the spectral lines allow you to understand the interaction between nuclear spins. And if you understand the interaction, you can reconstruct a three-dimensional picture. So having this type of spectroscopy, but now on a single molecule, will be extremely important for the field. But it is a hard task, and it's all about actually improving spectral resolution for having resolving the spectral line and improving the sensitivity to make experimental data acquisition time reasonable or even ideally faster than dynamics of molecules. So what you could then think about what you could do with this improved resolution in future, from detection of molecules, you can start to distinguish between alpha helix and beta sheet in the structure, or you can, by J-coupling, get information about tertiary structure of protein, and if you can detect a dipole-dipole interaction between nuclear spin, nuclear Overhauser effect, you can also have a quaternary structure of different subunits of protein resolved using this spectroscopy. But this is kind of still a big picture goal. I show you a few recent improvements and recent demonstrations of improving sensitivity and spectral resolution in this field. And improving sensitivity, it's about actually reducing noise, improving coherence time. So sensitivity is only dependent on coherence time, and coherence time is limited by environmental noise. So if you want to control noise, you typically protect your system either physically, so you in a cold atom experiment, you put your atom inside of vacuum and there is not so much environment, but this is not the case you can do for sensing. Sensor need to be very close to the molecule of interest. So the only thing you can now do is to protect actively using drive. And so a very conventional way to control the noise is apply a train of pulses. Similar, I was discussing before, these pulses rotate electron spin of qubits very quickly, and it averages out somehow external perturbations, and this works very well for static noise, or very low frequency noise. Many spin knockers are based on this type of application of a train of pi pulses. In general, this approach is called dynamical decoupling. If you use pulses, it's called often pulsed dynamical decoupling. There are also some limitation of these techniques. If noise has high frequencies, then you cannot put pulses so densely because pulses has some limited duration. So they are basically very well established pulse technique to cancel static noise, but when it comes to high frequency noise, there are some limitations. So another approach to realize protection of a qubit is to apply a continuous drive. Just to apply a microwave resonant with your qubit, and it creates a dressed qubit, and this dressed qubit is kind of protected by your microwave. So then there is an energy gap between dress states of your spin, and you need some energy penalty to flip this dress state, so it gets protected. And this approach is not requiring any pulses, you just drive your spin. It's called continuous dynamical decoupling, in contrast to pulsed dynamical decoupling, spin echoes that we have discussed before. So it seems to be a very promising approach because it's very simple to realize experimentally, so you don't need very short pulses and you don't need to adjust timing very well. But there is some caveat here. So if there is a noise in the microwave amplitude, that's create some kind of noise in a dress state qubit. So this technique is very sensitive to stability of your microwave source in terms of amplitude and in terms of detuning. And so far it was not applied very often. Actually, what was shown, what you can do is you can concatenate your decoupling fields. You start with one drive which protects you from external noise, and you create your dress state qubit, which is now a bit fluctuating because of the noise of the drive. And by applying a second microwave drive, which is much weaker, you can cancel effect of the first drive. You create a second dressed qubit, which is better protected compared to the first one, but it's still not fully protected because it feels the noise of the second drive, And in principle, if you want to cancel noise completely, you need infinite number of drives in this case, which is not very practical. So that's basically some of our limits currently, the performance of this pulse, of these continuous techniques. Recently, we have analyzed with colleagues at Hebrew University, Alex Svetzker team, special cases where the scheme can be simplified as many drives. It turns out that if first and the second drive have a noise which is correlated, for example, your microwave amplifier is having some gain which is changing in time, or your microwave source that generate both frequencies is the same, then there is a condition where the second drive can be detuned a little bit from the precise resonance, and in this case the effect of noise of both drives cancels, and there's negative interference of the noise. So there is a correlation between noise sources. You can put yourself in a situation where first noise get kind of a negative sign in front of the second one, and you can now, by choosing the right detuning, you can put a balance between these two noise sources, that there is a negative interference of the noise. So there are conditions for this, but for perfect correlation of the noise, you can find the right detuning, and then the noise will be fully cancelled, and this addressed state qubit will be completely stable. I show you here example of experiments. So where we detuned the second order drive, and I show you now Rabi flops, like a life of a qubit in time for a conventional single drive, and this is now decay of the qubit after a few hundreds of microseconds, not so bad, so similar to that we have seen before. And if you apply this double drive scheme with right detuning, the noises of microwave cancel, and the T2 time is going to about 2 milliseconds, so it's about order of magnitude better. And so reaching already limits that is given by interaction of this electron spin with phonons, some fundamental limit at this temperature. You can improve it by orders of magnitude, and that can be also translated now to better sensitivity of in-V-center magnetometers. We also work on other improvement of the technique very shortly. There's Flash-it. We also try to apply modulation of phases in this first order drive, and it's somehow very similar in terms of action on a qubit. Modulation of phases created in the field some sidebands, so similar to the second order drive, and it also can make your driving scheme very insensitive to fluctuations of power of the microwave drive and detuning. And so here are examples of how your qubit now in this driving case reacts on a single driving fields versus amplitude error and detuning, and these drives show that there's kind of a very high sensitivity, which is bad here, very high sensitivity to the driving field. And if you apply a right phase modulation, you get a very insensitive region, so your protection is very tolerant to fluctuations of the amplitude and of the detuning. Okay, and the second point that was important in context of quantum sensors is a frequency resolution. So frequency resolution is often related to stability of your clock and your internal clock of spin. In a very simple case where you use simple DC magnetometry approaches is given by coherence time of your color center itself. So the sensitivity function with this train of pi pulses have a width which is limited by inverse duration of measurements and inverse duration of coherence in the system. So it has some limitation in it. So your internal clock of your qubit is not very stable, and it gives you a limitation in resolving power in terms of frequencies. But in V-center, as we discussed before, do have a memory qubit, nitrogen nuclear spin, which do have a longer coherence time. So now we are working actually on employing this nitrogen nuclear spin as a more stable clock for NV center. So we call this technique based on nuclear spin correlation spectroscopy, and the idea here is that before starting the measurements, we initialize this nuclear spin in a well-defined state, and the electron spin is also initialized. After this, we run measurements with our NV center, accumulate the phase on the electron spin, and then swap it to the nuclear spin. At this moment, the nuclear spin is storing the information about the signal that was acting on NV center. And after the storage, the train of measurements is starting. And during the train of measurements, at the very end of the measurement, we measure the state of the electron spin and kind of add the phase that was initially stored in nuclear spin. And with this, we can measure the correlations between the phase acquired in initial period and the phases acquired during next period of time. Looks to be a bit complicated, but it's kind of now tracking the correlation of external signal over a long time, which is limited by coherence time of the nuclear spin, which is now longer. So now you can improve your resolving power. And so here you see the trace showing the oscillations of NV center as a response to external signal, and the time scale here you see it's about 30 microseconds, so it's much longer than this decay time of the electron spin of NV center by phonons, and it's now limited by T2 time of nuclear spin. So if you do Fourier transform of this waveform, you get a spectral line of external signal which has a line width of a few tens of hertz, so beyond the T2 time of the electron spin. Yes?
From my understanding, so you're introducing something like an artificial signal, right? And then with something that would be at the nanoscale, right? In the case of an
Yeah, that's right. So that was done for a single NV center, but that would work also for ensemble of NV center, and even if each NV center feels its own phase. So that's so it basically would work for NMR stochastic polarization. So exactly. So here it's basically it take a first reference points and then take second, third, fourth, until it goes through many points. Yeah. Okay, so this is currently the state of the art. So we can now do with this NV centers the detection of a few nuclear spins, but really big picture goal is to answer questions that biologists or chemists pose themselves about the structure and dynamics of molecules, and so further development of these techniques, and perhaps orders of magnitude still required to reach the sensitivity and spectral resolution level, which is sufficient to answer these questions. I was focusing more now on NMR, but NMR is not the only field where these qubits can be applied. If you are talking about magnetic fields and think about life, there are many processes in life where magnetic fields are involved. If the ions are passes channels, like for example calcium ion in neurons are flowing through channels inducing the action potential, they introduce in current which is about 10 picoamperes, and if you're very close to the channel, that corresponds to a field of about 100 picotesla. So that's something not very far from what we can measure with NV centers. Catalytic centers and enzymes making even larger fields coming from the electron spins inside of catalytic groups, which are on a microtesla scale if you are very close. Or neurons, when firing the signal, there is a current going through, and if you get very close to neuron membrane, so also the field estimation can be on the order of nanotesla. If you are more far away from the brain, so the signals are very weak, it's on a femtotesla scale. But if you get very close to membrane, it is expected that the magnetic field coming from the neuron signal is larger. And those are very exciting events, because they are very hard to measure or access with other techniques, except if you get magnetometers that you can approach to molecules to very, very small distance. And if you think about measurements themselves, of course you need to get close, and there can be different modalities. Either you take a diamond with NV centers created very close to the surface and you put a cell on it, and in this case you can sense something very close to the layer of NV centers, so perhaps membrane proteins or some events that are happening very close to a biological membrane. Or if you want really to go inside of the cell, you need to develop some other technique, and what currently community is working on is to use nanodiamonds, diamond particles carrying NV centers, and then functionalize it and then transfer it inside of cells. So this is now more challenging approach from material science perspective, but it's also more promising approach in terms of flexibility and applications in biology. So how to produce this nanodiamonds? So there are actually two ways to produce diamond particles, and you can produce nanoparticles by milling larger particles. It can be high pressure, high temperature synthesis of diamonds, so diamond powder first to 100 micrometer size, and then it can be dispersed, milled to a nanometer size. Also other approaches by producing diamond in the plasma conditions, in the chemical vapor depositions, and in some cases there is even self-nucleation of diamond particle in plasma, and they are falling down like a snow, having particles basically accumulated on the surface. Here I show you some examples of diamond powders of large scale generated by this high pressure, high temperature technique. You see the different colors. Actually, these colors are coming from different doping. This is color coming from nitrogen presence. This is after irradiation with electrons. The green color is coming from vacancies. And if you anneal this diamond, you get a pink color you've seen before for large diamonds. This is color of NV centers. So those diamonds carry these spin qubits, and now can be introduced inside of biological environment. Second important point, you need to functionalize your diamonds, so they should target some proteins. They should go to some place in cell of interest. And so there's another big effort in actually making suitable chemical groups on top of diamonds. And diamond usually is inert material, so it's not very easy to create suitable groups, and this is active field of research. I mentioned before amino groups that can link to some proteins. Carboxy groups also use it, and in some cases biopolymers are used to create a large number of groups where you fold your diamond inside of kind of biopolymer mass. There is also effort in coating nanodiamonds with silicon oxide, where the chemistry of surface is very well developed currently by chemists. So all these types of functional groups are possible to introduce, and then by incubating living cells and a kind of solution, hemming these nanodiamonds and then washing solution out, you can look with a microscope and now find bright spots inside of cells. Those are NV centers, and so you can track them. You can excite them using microwave antenna, which are shown here. You can do Rabi oscillations inside. You can repeat many of these experiments I've shown you before, now inside of living cells. Now, which questions we would like to ask? So, when I mentioned before, measuring magnetic fields can be an important avenue, but there are even more that the spin qubits can deliver. One of the possibilities is a measurement of temperature. Actually, temperature is playing an extremely important role inside of cell. It regulates metabolic processes inside of cells. It's even believed that temperature can be involved in the cell signaling processes. But measurements of temperature with high spatial resolution and high sensitivity is not trivial. It's not inside of cell level. Of course, microscopically, we can measure temperature very precisely. But if you come to a subcellular level, there's not so much methods that are existing. The golden standard currently is a temperature-dependent fluorescence. Some proteins, like GFP, has slight change in fluorescence intensity depending on temperature, change in fluorescence yield. And so the mapping of temperature based on fluorescence intensity is shown here. It's a GFP decoded temperature. And what was measured some time ago is that temperature gradients inside of cells, using this method measured, can be as large as a few degrees. So that created some discussion because that does not fit into model of thermodynamic of cells. If you take all the ATP that cell produce or uses, so you will not expect these gradients. You expect the gradient of a millikelvin. At the same time, the measurements show the gradients of a Kelvin. And this is still open question in the community, so we don't understand very well thermodynamics on a single cellular level. So the thermometer is very needed, and it's only one of the questions, so that's the most appealing question here. So now how the spins can be used for measuring temperature. We say we need a calibrated thermometer better than fluorescence. Fluorescence can depend on many stuff. It can depend on pH, not only on a temperature. So the spins are in principle not so sensitive to temperature, right? Because spin half system wouldn't feel the temperature, except maybe a relaxation kind of some. But for spin 1 system is different. So in V center has a wave function that is located around the carbons and the nitrogen. And because it's spin 1, there is a kind of intrinsic interaction between two electrons that creates so-called zero field splitting. So even in zero magnetic field, the V center states are separated by about 3 gigahertz. And this splitting depends on a confinement of the wave function. Now, if the temperature rises, diamond lattice expands, and there is a less confinement and less interaction between electrons. So that creates the dependence of the zero field splitting, so separation between 0 and middle between plus and minus 1 states as a function of temperature. Heat is measured by Dima-Budka group, and there is a shift of about 80 kilohertz per degree Celsius. The diamond lattice expands, that creates an 80 kilohertz shift per degree Celsius on the spin transition. Now you can think about whether it's big enough or not. So remember, it should be compared with coherence times. The lines are sharp at this scale, kilohertz, millisecond coherence time. And now you have a shift of 80 kilohertz per degree Celsius. That provides you an instrument to track the temperature with very high precision and very, very calibrated. In a bulk diamond, the precision of 5 millikelvin per degree per square root of hertz was demonstrated by your Prof group, but not with this precision yet for nanodiamonds. For experiment in cells, there was transfer of this technology inside of living cell. Here I show you the result of Lukin group, where embryo was doped with nanodiamonds, and what you see is a division event of the cell in the embryo, where you have these nanodiamonds which were introduced inside of cells on the right side, and they were used to track the temperature with a precision of about 1 degree. So these experiments really can be transferred inside of cells. They also can be applied not only by measuring temperature, but measuring some functional change of temperature. In collaboration with Tanya Weil group in Mainz, we have been able to introduce cancer drug on the surface of nanodiamonds, and the release of the drug was triggered by temperature. There were also molecules that absorb the light, get hot, and lead to release of the drug. That were kind of on the surface of nanodiamonds, and then we have been able to illuminate the cell, having these nanodiamonds as a bright spot here visible, and then measure the temperature by measuring zero field splitting, by measuring these spectral features, it shifts depending on temperature, and measuring this temperature basically was correlated with release of the anti-cancer drug. Maybe also one point about precision. So many experiments have been done by a very simple technique, just scanning the frequency and tracking the transition of NV center, but you can also do better using a more advanced coherent control technique. I show you here some special echo that is tuned so that it is sensitive only to frequency span between between -1 and +1 states and 0, and not sensitive to magnetic field fluctuations, creating shift of -1 and +1 kind of in a synchronized level. And this echo, which we call thermal echo, allow you to measure the coherence which is associated with like thermally sensitive coherence on a time scales of tens of microseconds, leading to the kilohertz line width of the transition. And that was done for nanodiamonds here for the first time. So the kilohertz narrow transition and the shift of spectral line by 80 kilohertz per Celsius, degree Celsius, allow you to measure the temperature very precise. They get on millikelvin precision level. Here I show you the measured temperature versus kind of temperature input in the cover glass where nanodiamonds align, and it fits very well on a straight line, and the error bar is already on a millikelvin scale of errors. All right, and maybe as a very last point, just in a few minutes, I think I'm a bit beyond the schedule, I also would like to show you that how the spins can be used for applications in MRI. So you have now nanodiamonds as an asset with the color centers inside, and they can be potentially also used for not only for sensing, but being object in imaging. So together with colleagues from Envision, our startup company, we have started to pursue the avenue where we take nanodiamonds, functionalize them as we discussed before, illuminate with light, we orient them the spin of NV centers, and then we transfer polarization from NV centers to nuclear spins present inside of nanodiamonds, for example, carbon-13 nuclear spins. So these nuclear spins are getting hyperpolarized, and then we inject them inside of patient or animal. At the moment, it's only possible to do an animal experiment and get much sharper, much stronger signal in MRI machine. And so I think I skip this and just show you the example of this that in micrometer size and submicrometer size nanodiamonds, we can increase the NMR signals, which is the basis of this MRI imaging, by a few thousand times for micrometer size diamond and about 1,000 times for a submicrometer size diamond. And this technique can be used in future for example, imaging nanodiamonds as markers inside of MRI. All right, I think with this I summarize again, so just a few points that were highlighted today. So now these NV centers are reaching the robustness and controllability to be able to detect very small magnetic field signals associated with NMR, nuclear magnetic resonance, like for example detecting single molecules, but further improvement of spectral resolution and sensitivity is needed. And what was not done so much in the community, but I think is very interesting, is to use actually entanglement, using not a single spin, but a couple of spins as a resource. And you have seen one example where nuclear spin quantum memory was used, but a variety of other scenarios are possible. For example, running quantum error correction on a few qubits here to improve the sensitivity, or use singlet state, which are decoherence protected, but are sensitive to gradients coming from outside. So another application is just hyperpolarization itself. So ability to polarize spins can be interesting for MRI imaging applications. And finally, I need to say that these demonstrations in research labs are not yet all that we need for real-world application. If you think about diagnostics or medical applications or applications of magnetometers in real world use. So integration of devices and making them user-friendly is very essential. And so there are first companies already started to work on this, making these diamond sensors to be basically on the way to market. And so first integrated devices are available nowadays. Right here I stop, I think. Thank you for your attention.
Thanks, Fedor. So, some questions, maybe? We have time for a couple, maybe. No specific questions from the students, maybe. I have one, of course. So, why the MB? So, there are no other defects in these solid-state devices that, that would make the same, more or less the same story, or is special or something?
Yeah, currently I think there are many other interesting color centers, but like a combination, unique combination of a few properties, like room temperature, long coherence time is not for many color centers. And this pretty high contrast in optical response to magnetic fields is also not for many. An ability to create them by ion implantation is not demonstrated for many color centers. So this combination, like being able to detect single with a strong optical transition, being able to generate with very high precision, having high contrast of optically detected magnetic resonance, so high response in percent in fluorescence if you apply magnetic field. This combination is currently unique for NV center, but maybe there are some others. Yeah.
Okay, from the point of view of the sensitivity and talking about the single MB, of course, the coherence time I can understand is something which, I mean, is very important, but also, okay, the readout of the photons, right? Because apparently to discriminate between 0 and 1 or 0 and -1 is also quite critical, I guess, no? Or, yeah, this is one of the things that probably is limiting sensitivity for the moment.
Yeah, exactly. So any loss of photons is influencing the sensitivity. Yeah. Collection is very important. So currently we are collecting just about a few % of photons.
So 100 experiments for collecting one photon. You said something like that, no? So
about this, yeah, exactly. That. So we are maybe like a few tens of experiments to get one photon out. Exactly. Yeah.
I see. Okay. So,
Thank you. Great talk. I mean, amazing work you've done. But I have a question. You have shown here that, okay, so MB centers can be sensitive to magnetic field, and also you have shown the sensitivity to temperature. What happens when you have multiple parameters present, the variation of both? For example, that is typical in biological tissues. I mean, you cannot make independent, the temperature variations from the magnetic fields of the cells. So how you approach this multi-parameter sensing with this kind of sensors?
Absolutely, that's very important. So fortunately, the effect of temperature and magnetic field can be decoupled to kind of orthogonal effect. So the magnetic field creates a splitting here between -1 and +1, but symmetrical. So they're basically moving apart, but the center between them doesn't move. That creates this delta here. But temperature shift -1 and +1 both together. So there is you can generate, if you measure two frequencies at the same time, you can discriminate effect of temperature when they move in one direction and the effect of magnetic field, which is created the splitting.
So that was the question. So you need to measure both.
You need to measure both. For example, it's in the simplest way, in this way, or you need to make a pulse sequence that cancel the delta and don't cancel this splitting. So this pulse sequence, which we call thermal echo, do exactly this. So it basically cancel effect associated with splitting between -1 and +1, but don't cancel effect associated with 0 to the center. To this.
So also in your work with Tanya Weil, so could you develop this a bit more? Because if I understood correctly, you put these nanodiamonds inside some cancer treatment or not? Or
Yeah, so it was loaded with a phototoxic drug, which basically get released from the polymer when it has reached some temperature. And so then the diamonds have been introduced inside of cell by endocytosis. So, and after this, the coating, which also respond to infrared illumination, so it can be brought to some temperatures, and then after reaching some temperature, the drug is released, and we can see this in measurement of phototoxicity of the whole cell line. That was not shown. And that was correlated now with the temperature that was observed inside of cell. So it was possible to correlate the action of the drug with the temperature of activation.
So maybe, I guess that one of the, what you want to do is to track more or less let's say the drugs, and to see in which parts of the body that can be located, or is different things. So
it can have different scenarios of application, exactly. So in a treatment level, so it's of course then a certain tissue area is illuminated where this temperature is reached, and there it is released, and it's not released basically in unwanted areas. So that would be then more in treatment cases, but here we wouldn't do thermometry with NV centers. But so here the question that was answered is to understand when the drug is released, at which temperature, and in a cellular environment, and whether it correlates with the action, with the toxicity. That was more for calibration of the action of the drug. So it was on a glass, so cells have been on a glass slide. Of course, we need to observe our NV centers, and that was more to understand, calibrate the action of the drug.
Okay, thanks. First.
Thank you, Fedor, for a nice talk. I was wondering, you talked about nuclear spin resonance. Is it possible, or would it be useful to do electron magnetic resonance imaging with the NV of a single molecule?
Yeah, I think it's also detecting electron spin attached to a single molecule. Definitely is something interesting. Yeah, it's require labeling then. So because all molecules carry nuclear spin, but not all of them carry electron spin. So usually you would require labeling and attaching. But it was also done by Jianfeng Du group in Hefei. So he detected proteins by labeling them with electron spins.
Okay.
So thanks for the talk, very interesting. I was wondering, particularly in biosensing, in biomolecules and cells, if you apply a microwave source, can you expect some absorption, and can you expect a heating in these cells? Can you expect some damage, maybe? And can this affect also your temperature measurements with MVs?
Yeah, that's a very important question, very good point. So there is some effect of microwave, and there can be two effects, like really microwave fields exciting water. So that was very small here, sort of, it's not directly in a resonance, and so we checked this as an effect. And the second effect, which is more like direct and more visible, if we put a microwave through the wire get hot, and so that relates to increase of a temperature in a sample. So we are trying to keep this duty cycle of microwave low enough to keep this effect under control. So in our case, this effect of laser radiation, we also need laser to read out, and microwave was less than a millikelvin affecting, but we have seen regimes where we put too much microwave, especially coming through the wire heat, and that was rising temperature by a degree or so. Yeah, that's,
Okay. I think no more questions, right? So, one more. Maybe the last one. Okay.
Hello, thank you so much. So when I'm curious about which region is measured when you have an envy. Normally there is like a dotted space around, but is that like you can measure inside and outside is not measured, or it depends on the distance, or which region can a single envy, measure?
Yeah, so you don't have like a sharp cutoff. So what it measures, if you're talking about, it measures always magnetic field. So it measures magnetic field at the location of NV. And now how far it reaches depends on a type of interaction. If it's a spin outside, then the coupling strength, magnetic field of external spin goes as a 1 over distance power 3, dipole-dipole interaction, or dipolar field. And it then turns out that in V center C, like most of the signal coming from the volume, which is comparable to the distance between NV center and the surface. So, for example, NV center is 5 nanometer below the diamond surface, or the size of nanodiamonds is about 10 nanometers. Then the volume that you measure will be about 10 nanometer across, 1,000 nanometer cube. So that would be the course. But there is still a contribution, a small contribution from molecules which are more far away, but this contribution is decay. So most of the signal is coming from the volume which is more or less the same as the distance between NV center and the surface, or the distance like to the sensing volume.
And inside of this region, every signal will be received similarly, or maybe the ones that are more close?
That's a very good point. So it's not similar because it's a dipolar field of the nuclear spin, so it has different contribution on different direction. And also in V center has a different sensitivities along different direction. It has a higher sensitivity for field along the NV axis. So it kind of have a radar which is more sensitive in one direction, and very low sensitivity for field perpendicular. So it has some anisotropic shape also in this. Yeah.
Okay, I think that was enough. So let us thank Fedor again.